Pronto, acho que ficamos aqui com a informação um bocadinho trocada. Eu não sei o que é que se passou, mas acho que foi o Moutinho buscou a informação do texto do e-mail, aqueles e-mails que nós fomos trocando, e não foi buscar diretamente ao Word. Depois houve aqui uma ligeira confusão, mas ficamos aqui com a... Mas acho que está um bocadinho trocada. Eu não sei o que é que se passou, mas acho que vocês estão a ouvir em atraso, não é?
Agora não estou a ouvir a Cláudia.
Desculpem, porque eu recebi uma chamada telefónica e ela não dá chance de responder.
Eu estava-te a ouvir bem, Eduardo.
Já estamos online, né? Já estamos online. Agora um pouquinho de silêncio, as pessoas já começaram a entrar.
Okay. Good afternoon in Portugal. Good morning, good evening, everyone, depending on where you are. My name is Eduardo Pereira, and I'm coming from ISIS and at the University of Minho in Portugal. It is my great pleasure to be with you here today in this networking Friday of Air Center. Today I am with Filipe Costa and Claudia Pascoal. They will be discussing around the topic of next generation biomonitoring and the sustainable management of ecosystems, the IBS perspective. I would like to leave you with a small note before we start. It's important that you place your questions using the Q&A button below in your window and not using the chat. Otherwise, we will not be able to collect your questions, okay? So without further ado, I would like to introduce you to Claudia Pascual. She will be introducing IBS for all of us. Claudia Pascual is a biologist and associate professor at the Department of Biology at the University of Minho in Portugal. She is currently the executive director of IBS and vice director of the Center for Molecular and Environmental Biology, CBMA. She was the former director of the Bachelor in Applied Biology and the Master's in Ecology at the University of Minho. Her research interests focus on assessing, predicting, and mitigating the impacts of global change on biodiversity and the functioning of aquatic systems. Over the past 15 years, she has coordinated several projects and both national and international, and established several partnerships with private companies and environmental agencies. Thank you very much, Claudia, for being with us today. I'll give you the stage for a short introduction of IBS. Thank you very much. Claudia, sorry, we cannot hear you. The micro is off.
Okay, thank you.
Perfect.
Thanks, Eduard, for the introduction. Hello, everyone, and thanks for the chance of being here and introducing the IBS, the Institute of Science and Innovation for Biosustainability at the University of Minho here in Portugal. So the mission of our mission in the Institute is to pursue sustainability in all its forms, protecting nature and promoting society development by bringing science and innovation to academia, industry, government, and citizens. We are highly engaged with the global goals for sustainable development, particularly these 7 out of the 17 goals. The biosphere goals are highly represented here. We have 2 infrastructures. We have a multidisciplinary team working in these 2 infrastructures. One is more devoted to biology and environmental sciences, and the other infrastructure, we have a team working in engineering and technology. So we have 4 research areas: biodiversity, ecosystems, services, and natural capital. The other one is resilience of natural and built environments. The third research area is space, oceans, coastal and inland waters. And finally, but the last but not the least, circular economy and bio-based solutions. We also have a business council composed of 17 private companies, very important players working here in Portugal, with whom we establish good partnerships, including a program of business and university shares. So we have currently 2 shareholders, one in smart systems for construction and the other one in sustainable communication networks for the digital society of the future. We also have an advanced training program. So here are some of the advanced courses we organized recently. In biodiversity, ecosystem services, river restoration, and sustainable construction. We also have we are also committed with stimulation of entrepreneurship, and we have together with the business sector developed some ideas and some prize for the best ideas to be implemented. And as a summary, I think I will keep this like that. Thank you.
Thank you very much, Claudia. I think, well, in any case, if you are interested to know more, you can contact Claudia anytime. We are very much looking forward to get your interests and your informations. Now I will pass to Filipe Costa. He will present his talk about Barcode of Life, or entitled Barcode of Life for New Generation Global Ecosystems Monitoring. Filipe Costa is a marine ecologist and associate professor at the University of Minho, Portugal, where he heads a research group on molecular ecology, biodiversity, and DNA barcoding. His research interests are on the development of high-throughput monitoring tools using environmental DNA metabarcoding, in particular focusing on estuarine and marine ecosystems, macro and meiobenthos and zooplanktonic communities. Filipe Costa serves as a Portugal delegate in the International Barcode of Life group, IBOL, in the Management Committee of European Cost Action DNA Aquanet, and in the editorial board of the scientific journals, Scientific Reports, and Metabarcoding and Metagenomics. Thank you very much, Filipe, for being with us today. The stage is yours. Thank you.
Thank you very much, Eduardo. Good afternoon, everyone. Thank you very much for the Air Center for the invitation. My talk today will have two parts. The first part will be a general overview of the relevance of the Barcode of Life initiative for the new generation of global ecosystems monitoring. The second part of my talk will address some new findings that we have on the molecular biodiversity, on the diversity of marine invertebrates in the Atlantic islands of Macaronesia. To accurately have data on species for their accurate conservation and management, we need accurate data sets of species occurrences and distribution. However, most of the diversity on the planet is still unknown to us. Most of it is what we call this dark task, is a vast majority of the life on the planet, and we know very little about this. What we know about the diversity on the planet is just a small fraction of this diversity. And even so, the means we have to identify and monitor this diversity are fallible. So far, we have been recognizing species and monitoring species using mostly morphology-based approaches. And there's a large body of knowledge that was built using these morphology-based approaches, which is very rich and useful for science. But there are some limitations. It's a low-tech procedure, and it's also a low-throughput procedure. So it would take hundreds of years to make it possible to inventory all the life on the planet, all of that 18 million dark taxa that we don't know about. So in 2003, a Canadian researcher, Paul Hébert and his colleagues, proposed a new universal system for species identification based on DNA sequences, and they dubbed this system the DNA barcodes. The DNA barcodes are thus short standardized sequences from a pre-established region of the genome that enable species identification in large blocks of life. And there are many advantages of this system compared to the morphology-based approach. It's applicable to any life stage or body fragment. It's relatively, comparatively faster and with much cheaper identifications on a large scale, and much accessible to people that are not experts, and also has lower uncertainty and ambiguity associated. So overall, this program will enable much simpler species identifications, accessible to anyone, even if they are not experts, and also will enable the acceleration of the biodiversity inventory, because there's so much life that we still have not described. The DNA barcode region, standardized region established for animals was the mitochondrial gene coding for the cytochrome oxidase 1 that you see here in this image. It's just a small fragment of this gene, and we only need 658 nucleotide bases of DNA to identify a species. Here you can see the DNA barcode sequence for a fly, and with this DNA barcode sequence, we can identify this organism anywhere on the planet where it occurs. So in order to have a global system for identification of all organisms, all multicellular organisms on the planet, we need a reference library where we can associate each DNA barcode sequence with a given identified species. So this reference library has been built over time since 2003. And what we need to build it is to identify morphologically one specimen from a known species, collect a tissue sample from it, extract the DNA, amplify with the particular DNA barcode region for that particular organism, and then deposit it in a public database. That effort has been done over the years, and there's a specific database where all of these DNA barcode sequences have been accommodated, which is the Barcode of Life data system, and which has already almost 10 million DNA barcode sequences for different kinds of organisms, from animals, plants, and fungi, and other species as well, other organisms. So altogether, we have implemented a system that is going to enable species identification at large scale on the planet and in an accessible way. But how is this system going to permit to speed up the species discovery? What we have been observing through the buildup of reference libraries is that the DNA barcode sequences cluster according to their similarity in a way which is a match with the known species. And for majority of species, there's a particular DNA sequence cluster that matches that particular species. And here you can see a dendrogram where for each different species, we have a different sequence cluster. However, sometimes what we find out is that for the same species, we may find more than one DNA sequence cluster. We call these DNA sequences clusters MOTUs, for molecular operational taxonomic units. And these sequence clusters, we have find out that they likely reflect true species. So when we find out MOTU that does not have a one-to-one match with a known species, that probably is an indication of finding a species that has not been described up to now. So in the scope of the DNA barcoding approach, all of these MOTUs have been attributed a barcode index number, which is a unique code that identify each of these MOTUs, so that we can recognize all of the species variability on the planet, even if they are not yet described. So this was the main development that we had on the DNA barcoding, as I described before. But a few years after DNA barcoding was proposed, DNA sequencing technology had a great advancement. There was a system that enabled high throughput sequencing, generating millions of sequences in each run. And this new system created a large opportunity for DNA barcoding to even have a broader range of applications. And the fusion of DNA barcoding concept with high throughput sequencing generated a new approach called DNA metabarcoding. So what is DNA metabarcoding compared to the normal DNA barcoding? So DNA barcoding applies to obtaining a DNA sequence, a standard DNA sequence from a single individual. But in DNA metabarcoding, we can analyze entire whole communities without sorting the specimens or even environmental samples of different kinds. So here we can see the basis of the DNA beta barcoding approach. So in this case, we do not have a specimen, we have a bulk community sample, and we can extract the DNA from this entire community, for instance, homogenating in a grinder, and then we obtain the DNA extract, which is a mixture of all of these organisms in the community. And then we can amplify through PCR amplification the particular DNA barcode region, and then use the high throughput sequencing platform to sequence all the DNA barcode sequences in that mixture. Then we have some bioinformatic analysis we have to do on the data here generated. And then we can compare the DNA sequences generated for that entire mixture with a reference database, and then get a taxonomic identification of all the species present in this community. So this opened new doors for environmental monitoring and enabled to apply DNA metabarcoding on a large scale for environmental monitoring, meaning that any ecosystem, terrestrial, marine, freshwater ecosystem can be monitored. Any kind of communities or plankton communities, for example, are very difficult to monitor. Water DNA extracted from the water, DNA extracted from the soil, DNA extracted from sediment, all of it can be analyzed for the presence of species. So this enables also a much higher density of the environmental monitoring in space, in time, and being able to generate a large amount of data on species dynamics on the planet. So to implement this large-scale new way to identify species, international consortium was established, the International Consortium for the Barcode of Life. Initially, there were there was an initial research program called the Barcode 500K, with the goal of generating the basal reference library with 500,000 species to initiate this new system for species identification. The current project is called BioScan. It involves more than 30 different countries, and it's going to extend until 2026. BioScan is one of the largest, if not the largest, biodiversity in the initiative in the planet these days. And the purpose of BioScan is to lay the foundations for a global system to monitor species dynamics using DNA metabarcoding procedures. So in order to implement this global system, it is necessary to have this reference database. Is absolutely fundamental. And so one of the major goals of BioScan is to increase this reference database from the 0.5 million that is currently, or it's already more than that, up to 2.5 million species. So more than the species we already know, with the expectancy that in the process of building up the reference library, we will find a large amount of undescribed diversity. So this was an overview of the DNA barcode initiative and its relevance in general for environmental monitoring at large. And now I'm going to show you some of the projects we have currently at IBS, particularly the applications of this approach to marine monitoring. The first project I want to show you concerns the usage of this high-throughput sequencing DNA metabarcoding approach for early detection and monitoring of non-indigenous species. The project called NIS DNA, and it's led by Sofia Duarte, and it's funded by the FCT, and also involves, in addition to the coordinator, the University of Minho, the University of Source, the University of Lisbon, and also with the support of the University of Guelph in Canada. So non-indigenous species are a very serious problem in marine environments, and we know that there are important vectors of introduction of non-indigenous species across the world, and among other there are the ballast waters from large ships, and also the fouling on ships and also recreational boats that transport these organisms across the world, where they may become invasive species and cause large environmental problems, and also with large economic costs. Morphology is not the ideal approach to try to monitor and detect these non-indigenous species, because it demands considerable expertise. It's a time-consuming process. The accuracy of the taxonomy is variable. In some cases, there's very low taxonomic resolution, meaning that many times we cannot identify at species level. And there's also very limited efficiency for for smaller organisms, and especially for some life stages like propagules, planktonic larvae and eggs, and these sometimes are the vectors of introduction of non-indigenous species, particularly effective vectors. So we wanted to devise a standardized DNA method for monitoring of non-indigenous species on the Portuguese coast. Our plan is to survey 9 different marinas across the continental Portugal and also in the islands of Azores and Madeira. But for the time being, we only analyzed we have only data for these 3 locations, Viana do Castelo, Porto, and Aveiro. And so what we did was that we went to these marinas and we sampled different kind of samples. Environmental DNA, so we extracted DNA directly from filtered water, zooplankton communities, and also the macrofauna associated with the hard substrates of the marina, like pontoons, buoys, and cables. Then we extracted the DNA from these different samples, and then we amplified 2 different genetic regions to have a better capability for detection through these DNA methods, DNA-based methods. And also we used then the IEEE sequencing over these amplified samples to obtain the DNA sequences. Then after obtaining the DNA sequences, we compared with the reference database. And these were the first very promising results that we had, still preliminary. But so far, in these 3 locations only, we detected 24 non-indigenous species. And you can see there's a huge variety of organisms. It would be very difficult to have a single person identifying these with great accuracy. So the system is working very good. And in addition to that, we had 4 new first records of non-indigenous species in Portugal that are here highlighted. So these results were very promising, and we want to extend the surveys to other locations that I told you about. And the document will be produced with guidelines for DNA-based detection of non-indigenous species. Another study that we have been conducting at IBS is focused on coastal macrobenthos. Coastal macrobenthos is very important in ecological terms, but also for monitoring. They are important indicator communities of the ecological status of coastal ecosystems. And they are very hard to process using morphology-based approaches. So we wanted to test DNA metabarcoding approaches in these communities and compare with morphology-based approaches. This was a work led by Barbara Leite and in a partnership with the University of Vigo, supported by the project NextSea and also FCT scholarship to Barbara. So what we wanted to do is to compare the colonization of artificial substrates that we deployed here in a location called Turana Island near Vigo. And these artificial substrates were then analyzed for identifying the species through morphology or identify the species through DNA metabarcoding. So we used basically the same approach that we already mentioned for the non-indigenous species, extract the DNA, amplify some different target regions of the genome, sequence, and then compare with the reference databases. And then we compared what we got with the morphology versus with the DNA-based approach. And these are our results. You can see different types of plates for artificial substrates that we used. And for each one, we have the global results comparing morphology and DNA beta barcoding based approaches. And as you can see, the number of species we detected with the DNA beta barcoding is much higher. And here you can see the split between different phyla of the different species that were found. And in fact, DNA beta barcoding detected 2 times more species and 2 additional phyla compared to morphology. So much higher capability for detection of marine invertebrate species. If we compare the partitioning between metabarcoding and morphology, we can see that most of the species were identified exclusively via metabarcoding. There are some also exclusively found through morphology, but in these cases, it can be some situations where the reference database does not have yet DNA sequences for these species. Some of them were rare and only occurred in one sample, and some other, there was some uncertainty about their morphological identification. So overall, we conducted additional studies following up these ones, and they all confirmed the DNA metabarcoding potential and efficiency for the high throughput monitoring of coastal macrozoobinthus. So to process a large number of samples in relatively short amount of time. Finally, one other study that we have in IBS is the fish DNA scan. This was a project that was funded through a specific call from FCT for the celebration of the 500 years of Magellan's circumnavigation voyage. So it was important to involve countries that were on the route of Magellan. And so we are a multi-country consortium involving Portugal, Cape Verde, and Brazil, these 3 countries with, in the Atlantic to use this DNA-based approach to look for fish diversity. I'll tell in a minute what it is about. There are several institutions in Portugal, Cape Verde, and Brazil. In Portugal, we have University of Minho, Coimbra, Algarve, and also IPMA is involved. And in Cape Verde is the AIMAR, and UTA also are involved. And in Brazil, it's the Universidade Estadual de São Paulo. So this is a large consortium with the goal of using DNA-based approaches for species identification, in particular for ichthyoplankton, meaning fish eggs and larvae. And ichthyoplankton are a particular useful target for this kind of DNA-based approaches because it's very difficult to identify species of larvae of fish and even more difficult from fish eggs. So we intend to conduct a proof of concept study of the application of this approach in the 3 partner countries. And also we want to explore the potential of using seawater environmental DNA as a non-invasive approach for monitoring coastal fish species. So ultimately, we want to use the data that we can produce using this DNA-based monitoring to get data with high spatial and temporal density that can support fish stock assessment and fisheries management, namely by helping to identify spawning areas, spawning seasons, intensity of recruitment simultaneously for multiple species and with high spatial and temporal density. So these were the applications we have seen for marine communities, different types of marine communities that we have on IBS. But DNA metabarcoding is applicable to all communities and ecosystems. And in fact, there has been a network that has been created in Europe called DNAquanet, funded by the Cost Action. And this network had precisely the goal for developing genetic tools, DNA-based tools for bioassessment of aquatic ecosystems in Europe. And recently, it was launched the European branch of Bioscan, and this will then address all kind of ecosystems using a DNA-based monitoring approach to monitor species dynamics in terrestrial, marine, and freshwater ecosystems. So now we finished the first part of this talk. We move now to the second part. And in the second part, I want to come back a little to the first stage of DNA barcoding before metabarcoding, because we use this approach, the single specimen DNA barcoding gathering. And through this approach, we have important new insights on marine invertebrate biodiversity to show you that we found in Atlantic islands of Macaronesia. Macaronesia is a very interesting region from the biogeographic point of view. It's the assemblage of these different Atlantic islands, Azores, Madeira, Canary Islands, and also Cape Verde. Cape Verde is part of it, although in our study we did not include Cape Verde, unfortunately. All of these islands have a volcanic origin and never had a connection to mainland. And they emerge at different geological times. The oldest one perhaps as far back as 30 million years ago. The most recent ones about 300,000 years ago. Anyway, these are old islands. And it's very well known that numerous endemisms are common in the terrestrial fauna and flora of these islands. And this has been promoted by colonization, followed by geographic isolation, plus divergence of these endemisms. So this is a very well-known sequence of events that is well reported and well known. However, for the marine invertebrates, little is known about island biogeography and evolution. And particularly, so far, we have little data on marine invertebrates. And because there's no apparent barriers to dispersal in the open ocean, marine invertebrates in these islands have been assumed to be identical or very, very similar to nearby continental coasts. So there's a faunistic continuity between these regions so far with the information we have. So in order to get to know a little bit more about the diversity of marine invertebrates in these islands, we conducted a series of studies that I'm going to show you. This one was led by Pedro Vieira and was focused on the small crustacean, Dinamen edwardsi, which is very common in the intertidal regions, has a prominent and important role in the ecosystem. And we collected samples from different locations, including the Mediterranean, the Portugal, Azores, Canary Islands and Madeira, and also in Morocco. And this organism has a particularity, which is the fact that he has direct development, meaning that he does not create larvae and he does not disperse through larvae. So possibly this can be an organism with lower capability for dispersal than other organisms, but this is a putative conception that may not be realized in practice. However, when we analyze the DNA barcode sequences for this organism collected in all of these different locations, what we find was this phylogenetic tree that you see here, and each of these branches actually is a different, very divergent genetic lineage that we found in different locations of the populations of these organisms. So it looks like different populations actually may be, in fact, we have 9 different MOTUs, sequence clusters that we found. And we have a number of additional data that indicates us that these are very likely separate species. of these organisms. Additional main findings, these are deeply divergent. The genetic divergence between them is very deep. So we are very safe that these are different species according to all the information we have. And curiously, they are also geographically segregated. As you can see here, you see the Motu from El Hierro is separated from La Palma, from Morocco and Selvagem, and these are all islands very close to each other. And notably, 6 of these different species occur exclusively in one island only. So as far as we know, this is the only known occurrence of these species in the planet with the current data. We went on to check another organisms of the same kind, also lacking larvae stages. And this is another group of crustaceans, the amphipod crustaceans. And we analyzed in a work done by André Desidrato and Pedro Vieira, 7 different species of the family Hyalidae. And we collected samples from many locations in Macaronesia, also in Iberia, but also in other countries. And then we analyzed the partitioning of the DNA sequence data from these different populations. And we also have here the results of our findings. Each of these branches that you see here in this phylogenetic tree are a different MOTU, so they are probably a different species that we found. And altogether, from these 7 different species that we analyzed, we came up to between 26 to 32 MOTUs, depending on the method of clustering sequences that we used, but still much higher than the 4 to 5 times higher than the species. And an important finding was that most of the MOTUs of the same morphospecies were geographically segregated. So each MOTU occurred in a different location. Most of the MOTUs were endemic to Macaronesia, which is also an important finding. And also the genetic diversity within these MOTUs, the variability and the DNA sequences also showed that the highest diversity was also in Macaronesian populations. So we have additional evidence from other group of organisms for the occurrence of high diversity in Macaronesia marine invertebrates. We were not still happy with it. So we started to think maybe this is a pattern that is common to all of these kind of paracarid species. So we decided to go further and analyze 23 of these species that we knew there were populations occurring in Iberian Peninsula, in the Macaronesian Islands, and also in Morocco. And when we analyze all of these groups of organisms, you can see here that the populations from Iberia are in red and the populations from Macaronesia are in green or blue. And you can see that there are different branches for each of the populations. So in most of the cases, or all of the cases, there was a split between the populations of Iberian Peninsula and the populations of Macaronesia, and high levels of genetic divergence between these different populations. So here in this graph, you can see the density of taxonomic and genetic variability according to the red color. So you can see that there is this true hotspot for these species that we analyzed in Macaronesia of genetic and taxonomic diversity compared to the other locations. The composition of species is similar between the islands of Madeira, Azores, and Canaries, and is also similar between Iberia and Morocco. So there's a split between the island and continental species. We found out actually over 60 new suspected species in this analysis, and 57 of these species are endemic to Macaronesia. So it's the only way we know that they occur. So there are important biodiversity implications that I'll talk later. Now, the question for us was, is this a pattern that only occurs in this particular kind of organisms, or can we find this in other type of marine invertebrates, namely those that have a pelagic larvae, like these polychaete annelids that Marcos Teixeira analyzed. He analyzed 3 species from these locations that you can see here. Also, he analyzed some from the Mediterranean and from the Macaronesia and Iberian Peninsula, a project sponsored by NextSea. And these are the results for one of the species. In this species, he found 14 different molecular operational taxonomic units. So 14 different genetic lineages. And we are very confident that these genetic lineages correspond to different species. We use different molecular markers, and we also had large distances between these lineages. So we are confident these are different species. And you can see there's a high density of different species here in the Macaronesian Islands. In fact, the summary, and I'm almost finished if I'm taking too long. In summary, this analysis for these 3 polychaete species, these were the results we got. So here you can see the number of MOTUs or genetic lineages found in each of the locations analyzed, and then between parentheses, the ones that are endemic. And you can see that here in Macaronesia, there's a very high level of endemicity. Just with these 3 species, we have 9 exclusive MOTUs for Macaronesia. So altogether, we seem to have found DNA-based evidence for a marine biodiversity hotspot in Macaronesia. We found this in 2 groups of organisms, mostly on these paracharid crustaceans, but also on a few annelid organisms. We want to highlight this, that these findings, it was essential to use DNA barcodes, and they will be essential to detect and to monitor this diversity. However, we want to further confirm this pattern in other taxa, in addition to these two taxa, and more within Annelida for sure, and also supplement these findings with other approaches. Anyway, so far, we have found numerous endemic species in Macaronesia, and they have very narrow ranges. And very narrow distribution ranges means that these are much more susceptible to population declines and even to extinction because they have very narrow ranges. And in fact, this is a unique taxonomic and genetic diversity heritage of marine invertebrates that we found here in the islands of Macaronesia. And probably this is the only place in the planet where these unique genetic lineages occur. So in terms of biodiversity and conservation and management, we must take this diversity into consideration. For example, if we want to design marine protected areas, we must consider the fine scale island-specific endemisms in the design of these areas. One final note, just to show you the map of the ecologically and biologically significant marine areas classified by the Convention for the Biological Diversity. And I find it curious that Macaronesian islands are not classified as a EBSA. And I hope that with this new data, maybe there could be a reclassification of this region also for marine invertebrates, because so far it's only recognized for the endemicity of terrestrial fauna and flora. Therefore, finally, I wanted to acknowledge the researchers and students that contributed to the data I showed you here today, to acknowledge our sponsors, FCT and North Wind, and also the Assemble program, to thank other colleagues that somehow had a role in this presentation, and also to the International Barcode of Life, Bold Systems, and the Anaconet. And thank you for the Air Center for the invitation, and thank you all very much for your attention. Thank you very much.
Thank you very much, Philippe, for a very, very interesting presentation. It was very nice to follow it. Well, I think we have many, many questions. I'm not sure if we will be able to discuss all of them in this session. In any case, I will just leave this reminder We will collect all the questions and we will try to address them all, even if it's not possible during the session. After the session, we will be making these answers available. I will start with Omnia Ibrahim. He was very active with many questions. He asked us, well, first question is the PCR and the identification of barcodes any help in the identification of all taxa? So this is a general question, but in particular is interested also in knowing if it's possible to use barcodes for oysters, or if you have any experience in identifying oysters using barcodes. And also I will just add another one because he has many questions. If for example, if it's possible to use this technique for specific genes or for infectious diseases, or in case of polluted areas, if it's possible to have any trace on this type of identification technique in the DNA material.
Okay, okay. Regarding the organisms, in principle, you can apply it to oysters for sure. And in most of the cases that are known, only with a few exceptions, most of the organisms can actually be clearly discriminated using these DNA barcode sequences. And I'm talking about multicellular organisms and mostly about animals. There are specific DNA barcodes established for plants and specific DNA barcodes established also for fungi. In the case of plants, the discrimination may not be sufficient some of the times. It's a bit more difficult and we need more DNA barcodes for discriminating plants. But in general, for animals, it works pretty well with a few exceptions, like for instance, in cnidarians, sometimes it's not so easy, and some other groups like Porifera may be a bit more difficult. But it is a very universal system that can be applied to almost any organism. Regarding the technical issues, I don't know if I could understand exactly what is the question.
Yeah.
If this technique could be used to detect organisms in environmental, basically the concept. The concept of DNA barcoding can be applied in any biological system, basically. You have first to determine if your DNA barcode sequence is able to discriminate the target species that you want. And then eventually, if you have a specific species, you can develop a specific protocol to detect that species. The concept will be the same, although the DNA barcodes, the logic is to use it as a universal marker to monitor species in the environment in different conditions. But the concept can then be applied to other regions of the genome as long as you have a reference library that you can match your unknown specimen to the reference library for that particular region of the genome. I hope I was clear enough.
Well, Omnia had many more questions, and he has also a question on, well, how the researchers can join IBS. I guess he will be interested in exchanging some more ideas with you. And
feel free to contact me. I'll be glad to help.
I will try to address some of the questions that others have posed. And if there is time, I will come back to the questions of Omnia because he had many. Jerome Benedict, he also is asking, are there any ongoing or planned collaborative works in Asia Pacific region already, as far as you know, Philip?
Well, within the BioScan project, there are major countries in the region that are involved. And I don't know specifically projects that are being developed there, but I'm sure these countries have their own networks for barcoding internally. And I don't know of any project in the region, but you have China, you have Australia, you have New Zealand, you have, and probably more countries will join the BioScan effort in the future. So one way to do it is you can go to the website of BioScan and try to make a contact and see if there are any particular programs for that region of the world, or if they are at the moment only within each country. Anyway, this is a global project, BioScan, so it's going to cover all regions of the world and all kind of ecosystems anyway.
Okay, thank you, Philip. Abubakar from Nigeria also leaves a question that is going more or less in the same direction regarding eyeball. The map that illustrates the 3 research programs only shows 2 African countries, South Africa and Nigeria, barcode 500K between 2010 and 2015. And he's asking if you know, if there are any plans to include other African countries, and if you are aware of any barriers that hamper a wider participation from Africa.
I think there are no barriers really. I think as long as there are strong institutions and strong participants in these countries, meaning that they can have actual contribution to the program, I'm sure they can be involved. And if they have such a system that they believe that they can have a good contribution to this program, it's a matter of contacting BioScan, going to the website, contacting BioScan, and try to engage in the program. This is the goal, is to be a global initiative. So as long as countries have the conditions to participate, they are able and they are welcome to participate. I'm not leading the BioScan effort. I'm just talking as far as we know. As far as I know, from my position within the initiative, but I think it's very welcome for everyone to join.
Yeah, thank you, Philip. I think there is a lot of interest around the eyeball, and we have some questions. Okay, now also from Nigeria, Musa is leading the following question. I think you maybe addressed it in your presentation. How many species can be found in eyeballs database today, if you know, and if there is any taxa that are more relevant or prevalent in the database, and what's the proportion between terrestrial and marine species? I think, well, in any case, she leaves a comment. It was a wonderful presentation.
Thank you.
She thanks it, she also asks if it's deployable in African universities. So what are the technological tools or means that you need to conduct this type of research? I think, well, this is the question.
Okay, so regarding the amount of data that is currently available in the reference library, to this point, we have over 750,000 different barcode index numbers. So these are not species, but they are proxy to species. So they are probably, in fact, we have that number of species in the database. It's true that most of the effort has been allocated to terrestrial animals. There are a bit also of plants and fungi. And within the marine, I have been one of the persons that have been contributing. There's been various contributions around the world, but I would say that marine organisms are a bit slower, going a bit slower, the population of the reference library compared to the others. but it's already a very large number. And with this Bioscan program, the goal is to increase and cover all the known biodiversity on the planet. Regarding the application in Africa, there are different ways this can be done. One way is to try to, and that this would be the optimal way, to implement the technologies in African countries, but these are expensive technologies and would require some training for the people using it. But if there is funding available for that, it would be the ideal situation, because then each country in Africa could contribute their own specimens and process their own specimens in the location. However, there are big hubs of DNA sequencing for this project in the world, and in Canada there is one of them. And if another possibility is African countries to send their specimens to these big sequencing hubs and then receive back the information about their own diversity.
Okay, thank you very much, Philip. Another question from Jose Mulbert. He asks, well, basically introducing, he says that to use environmental DNA or DNA barcoding in an applied manner to fisheries, for example, you need high resolution, time, and spatial sampling, which will result in a high number of samples. What is the feasibility of doing this time of analysis at the moment, costs and infrastructure? So I think if I can summarize, what is the feasibility of using this technique to apply for fisheries, maybe for abundances and for species identification? What's your view on this? Costs and infrastructures also, by the way.
Well, currently we still don't have sequencing technology developed for in situ processing of samples, or it would require, for example, if you go on a survey, of course, you can take some of this sequencing technology with you and process the samples there. But most of the work so far has been done in laboratories. So usually what we do is that we collect the specimens in the field and then we take this to the laboratory and do the extraction. If you have a possibility to go on a big vessel which have laboratories, then you can implement this DNA sequencing technology within the research vessel, and you can process many different samples. The costs are a function of scale. So the more samples you process, the lower are the costs at the end. So if you process only one or two samples, the cost will be high. But if you process many different samples, then the costs come down. The trend in the sequencing technology is to become more and more portable. So I guess in the near future, it will be much more easier to do this kind of analysis in situ. So it's a matter of time until these approaches can be readily available for large scale campaigns.
Okay, thank you, Philip. Thank you very much. We had many more questions to answer, but unfortunately we have to move on.
Thank you all for your questions.
Try. Yeah, we had very nice feedbacks. We will maybe collect them all, and then if you can reply to them, and then we make them available to all of them. So with this, we move on to the next speaker, to Claudia. She already gave an introduction to IBS. Now she will give her talk on biodiversity and climate services towards agri-food and water sustainability. Thank you very much, Claudia. The stage is yours.
Thank you. So first of all, thanks again for the opportunity of sharing our ideas and our work. And congratulations for the initiative of this Friday networking that are really a big success. So today I will talk a little bit about bioeconomy, biodiversity in the sequence of Philip's talk. and climate services. And I will have a focus on the agri-food and water sustainability. So first of all, I would call your attention to the world population growth and projections. So nowadays we are more than 7 billion, and within this century we will attain for sure, even in the lower projections, about 9 billion. So there are contrasts. We are aware that there are contrasts in world population projections according to the region. For instance, the shares in percentage per Asia are always the great majority. Europe is decreasing and Africa is really, really increasing. So the big question is, where will we find enough food for 9 billion? By other words, how can we feed the world? Of course, we can think about expanding our agricultural area, but this has been expanded a lot over the last years. So I think there is few available land. I think we can no longer afford to increase food production through agricultural area expansion. So we have to think about alternatives. For instance, increasing farming in cities. For instance, growing food locally. For instance, increasing farming in cities, growing food locally with vertical farms might be an option. But there are also other threats on agriculture. For instance, the transboundary old and new pest diseases that are threatening agriculture. And one of our focus is on the link with biodiversity. We are convinced that enhancing biodiversity will increase ecosystems' resilience against diseases. So it's one of our major focus. But there are also other challenges and other threats. For instance, climate change. Climate change is affecting agriculture with implications for global food security. And this is really complex and needs a very integrative approach. So our approach is to use a circular bioeconomy strategy based on sustainable development goals, in which the biosphere-related goals really need to be protected because they are the basis for all the benefits, for all the society and the economic benefits that we can take. So this was the rationale of a project that I will present to you, the Eco Agri-Food project. It was a large project developed by here at the IBS, and it intended to develop innovative green products and processes to promote agri-food bioeconomy for the sustainable development and towards business competitiveness. So our major and last goal was to lower the pressure on natural resources, to adapt to climate change, to enhance human well-being, and to promote biosustainability. And we did that by using innovative microbial cell factories. So we used microbes for producing new products and processes together in close cooperation with the private sector, with the food industry, and exchanging the technology and the knowledge with these companies. And we also had always the awareness and the awareness of the human and environmental risks of these new products and processes. So we are always aware of the biodiversity and ecosystem health. Of course, biodiversity and ecosystems are also sources for new products that can be used for producing new products and new processes. So this is quite a cycle that we have to be aware. So for instance, we have been developing in this project new peptides, antimicrobial pesticides, enzymes, and food additives that are very useful for the agri-food sector. And these products should have a track on the ecological footprint. So our aim is to reduce the ecological footprint of the agri-food sector. Of course, we have been exploring biodiversity and biological resources for the valorization of biological wastes. that the agri-food sector is producing in big amounts. And as using the technology that Philippe explained to us, we have been also applying next generation biomonitoring for biodiversity assessment in different ecosystems. And we have been mapping in this project biodiversity in agroecosystems and use biodiversity as an asset to support ecosystem services. And we have been noticing that promoting biodiversity and increasing ecological complexity is enhancing productivity and is enhancing and improving the resilience against pests and diseases and against climate change. And some of the projects, some of the interactions that we had with our stakeholders are related to the wine production. So we had here some of the outputs that were clearly communicated in the national press. But of course, this project, I think we have been advanced a lot in terms of knowledge and interaction with the companies and with the agri-food sector. But we know that there are a lot of challenges that we cannot address immediately. So there are still a lot of risks, and we have here the global risks for 2021 reported by the World Economic Forum. And interestingly, five of the major risks are related to the environmental stuff. So in this graph, in our right, we have the likelihood of the risks increasing from the left to the right, and the impact of the events increasing from the bottom to the top. So in this square on the top right, we can see the major risks for the future. And as you can see here, the failure of climate action is really one of our major threats. And considering these, among the climate action and the risks of climate failure actions, we have water scarcity. Indeed, for agriculture, water is a major we have a high dependence on water. And here we have a map with the water scarcity in major river basins worldwide. So we realize that there are already regions with a lot of water stress. And this is important because globally, 70% of fresh water is used for agriculture. It's not equally distributed in all countries and in all continents, but in average, it's 70% of fresh water used in agriculture. So we think that with the increase in population growth, with the increase in the agricultural needs, there will be an increase in the water stress. And not only for agriculture, but also we have to look to the water footprint. So here we have food is being produced in one country and uses the water of that country, and then there will be the trade of the industrial and agricultural products to other countries. So there is a trade of water as well. So here we are representing the net vital water import from one country to the others. And if water scarcity is on our way, we think that water stress will increase. And is projected to increase as time goes by due to water scarcity and increasing water demand. So we are aware that conflicts because of water may occur in the near future. And this was exactly the rationale of a project that we are coordinating here at IBS, the ClimateAlert project. The ClimateAlert project is developing a ClimateAlert smart system for sustainable water and agriculture. And the main goals are to contribute to improve the transfer and information on weather forecasts, together with near real-time satellite information. And we aim to develop tools to incorporate climate information in an easy way, useful for decision-making namely in the agriculture and water sectors. And we aim to mitigate the impact of extreme events and to develop an adaptive management attenuating the conflict between the water and human activities, namely agriculture. And we did this with a consortium with different expertise, as we usually do it at IBS. So we joined people from social sciences, at this time from Germany, from UFZ. We joined climatologists and hydrologists from IPMA in Portugal and also from UFZ in Germany. And we joined a team of engineers from our IBS here in Braga and in Guimarães at the University of Minho. And we joined 2 teams of ecologists here at IBS and also in Catalonia, the ICRA team. And we joined these people working together, and we selected 3 case studies. So one case study was in North Portugal, in the covered river basin, and where the main pressures in here, we can see the main pressures are agriculture and urbanization here in North Portugal. We have a river basin with hydromorphological alterations. We have a high complex of dams interfering with the system, with the natural system. We are very prone to winter floods, and we are also prone to summer drought events. Then we have another case study in Spain, in South Spain, the Algarve River Basin, where the main pressures are related to agriculture and livestock. And the river basin is also impacted by some chemical alterations, minor hydromorphological alterations, but major chemical alterations due to agriculture and lack of water. So one of the major threats is related to drought with impacts on agriculture and in the river basins. So the drought is so severe that a big discussion is going on about what shall we prioritize, to give more water to the people or to get alternatives to water supply to humans, and to contemplate also the needs for agriculture. So there is a big issue here on that. And also the third case study was in Germany, the Saal River catchment, where the main pressure is agriculture. It's a high intensive agriculture area, very prone to flood events. And interestingly, over the last year, they were prone to summer droughts. So people in this area are now very concerned about climate change and climate alterations, with the impacts on their not only in their business and agriculture, but also in their daily life. So based on these 3 case study areas, we aim to develop 2 tools, 2 climate service tools, one mobile app more devoted to farmers, and a web application more devoted to water managers. And to develop these tools, we had to interact with people and with our stakeholders. So we had several workshops and inquiries for engaging with civil society and policymakers. So we had to listen to people and to co-develop the tools with them. So we came up to a list of indicators that people wanted to have information on that. So we had to listen about what kind of indicators people wanted, how was the frequency of that information that could be useful. And then we had to think about how can we access, where is the data, how can we use the data, where is the data, how frequent is the data, and how can we communicate data in an easy way. And we had data that was from our national meteorological agencies, but also used a lot of data coming from the Copernicus service in Europe. And we came to the Climate Alert mobile app here, so we can have information on soil moisture, on vegetation index, on precipitation, on surface temperature, on evapotranspiration. So people can select what they really want to measure in their area of interest. I would like to have a demo here. I would ask Katarina if she could help me to show a small demo of the app. Can we listen to Katarina? Could you share the the website. Okay, so here is our app, if we can, okay. So we opened up, it's loading the data. So we have the information, we can select the language, and we can select our area of interest. We can switch to a different view, and we can select the indicators that we want in the selected area. This is a farm where we have a stakeholder interacting with us. We can change the background if we want, and we can select the variable we are interested in. For instance, for precipitation, we can see the precipitation for the last days or for the next few days or for the current months or the previous months. So we can compare with the normal for the period. We also can compare with previous years. So as an agriculture, as a farmer, I can compare what is normal for the year. I can compare with the previous year, and we can also provide seasonal forecast for the next 6 months. Of course, we know that there is a lot of uncertainty in this seasonal forecast, but we provide the best available data we get so far. And this has been quite useful and have been used by our stakeholders in the 3 case study areas. And now I can share with you okay. So this is the app, the mobile app that is really more devoted to the short-term decision making. So people, farmers can think about, make a decision on, shall I irrigate today or tomorrow, or shall I anticipate my harvest, or shall I add fertilizers or pesticides? So according to these projections, I can anticipate a little bit my actions, and I can save a little bit money without saving water, saving fertilizers, or saving money invested in pesticides, and I can save the environment. So the idea is to get productive crops, but saving money and saving the environment. Then we developed another tool, a web tool, and this web tool is for medium term and longer term predictions. And here we provide information on annual data, wet months, dry months, at the short and long term, related to ecosystem services, related to water provision or water quality and water purification provided by the river basin. And we can have a zoom in the sub-basin scale or at the ridge scale, and we can provide information also in graphics with the projections for the short term or for the long term. And we can also incorporate here and show to our stakeholders all the projections, including the models used and created by ICCP. So with these projections, what we provide to our stakeholders is the ecosystem service provision related to water stocks, water quality, and we provide that information for our current status, and we provide the scenarios for the next 20 years or for the next 40 years using different climatic models. A climatic model that is a bit more conservative in terms of reducing the greenhouse gas emissions, or other scenarios in which we predict to have higher concentrations in the future of greenhouse gas emissions. But it's useful for if we want to planify, if we want to make investments in what kind of crops we want to have in the future, what kind of investments we will have in the future. If we have a particular area that will be particularly susceptible, particularly sensitive to water constraints or to temperature constraints in the future, we have to have that into account if we want to make investments. So this is our two tools that we have been developing very, very close to our stakeholders. And of course, we have just the tools we're operating now in 3 different macroclimatic areas in Central Europe, in the Mediterranean area, in the Atlantic Europe. But we have different climatic pressures from droughts to floods in these 3 regions. And of course, if we can have access to data, satellite data that are very useful for making these tools operating, we can validate this transnational system. And this will open an opportunity to later transfer these climate alert tools to other countries facing similar climatic threats. And of course, these two projects I've shortly explained to you were only possible because we have a very enthusiastic and very committed team at IBS operating in collaboration with other countries and other partners, either in Portugal or abroad. So thank you very much for your attention, and I will be pleased to answer to any questions you may have.
Okay, thank you very much, Claudia. It was very, very interesting and very, very important topic. Both projects are very much related, right? There is a very good interrelation between both. I have a question, if I'm allowed, since we are still waiting for some questions. Well, basically, my question is, in your opinion, if it's, well, you addressed all these issues during your presentation, but in your opinion, what could be the greatest challenges that you may identify for agriculture in the near future? Some examples, which ones do you consider more important, or food production, right? Is there a lack of information, or is it possible to get more information, or could this be a solution, or is the population growth the greatest challenge? Are plagues or climate changes, water stress? What is, if you could identify the greatest threats for sustainable agricultural or food production, what could be those the major ones?
Yeah, thank you for the question, Edward. For sure that the increase in human population will be a major threat. How can we feed all these people? It's already a threat already, and it will increase in the future. But if we need to produce more food, we have to think how can we do that? And we cannot occupy much more space for agriculture. So we have to think about alternatives. And the alternatives, I think they should be related to increase the efficiency of the process and to have in mind sustainability, because agriculture has to be sustainable, and we depend on that. But at the same time, agriculture will depend on water and temperature. So the crops are highly dependent on water availability. And as I said before, 70% of fresh water is used by agriculture. It's incredible. It's much more than industry. It's much more than domestic use. It's a huge amount. Agriculture and livestock as well. But it's an incredible number. So we cannot avoid it, because we have to be aware that agriculture really needs water, and no other chance. But of course, we have to save water as much as possible. So I think we should move towards to provide and to use the water in minimum amounts as possible, without taking any risk for the production, of course. So a smart system, it's the ideal system. So we should not irrigate today if I know that within the next 3 days will rain around. And sometimes we don't care about that. We just think that we have to irrigate the system, and by default, it's being irrigated. So I think that's an important point. And also, if we are using fertilizers that sometimes are useful and very important, and sometimes we are using pesticides because they are also needed because to avoid pests and diseases, we have also to use the minimum amounts, because I can save money with that. If I save water and I have no doubts that the price of water will increase in the near future. I read very recently the newspaper, either in New York or even here in Portugal, that water is becoming more and more a commodity. So probably in the near future, I wouldn't like to have that, but probably in the near future, we'll have water in the market, in the financial markets, like the gold or the oil. I wouldn't like to go in that direction because it's an essential and vital resource. But probably we have to be prepared that if the water stress increases too much, we may move towards that direction. So of course we have to save the water as much as possible. Of course, the cycle of water means that it doesn't end. Water is always there. There is no beginning and no end. The water is here. And sometimes people think that is always available, and it is not. Water scarcity is a reality in many regions. And sometimes in a very short term, it rains a lot, but we cannot use that water because it rains in such a short term that we are not able to benefit from that available water. So we have to have in mind that agriculture needs water and we need to feed the people. So we have to use in a very rational way. And with the climate change, scarcity will for sure increase, and temperature will increase as well. And with increasing temperature, of course, we have to adapt. There is no chance. We cannot fight against nature. We have to try to adapt and to cooperate with nature. So sometimes we need to move a little bit in altitude in terms of agriculture strategies and activities, or we have to change the crops, or we have to select the best crops adapted to my climate and not to force in the other way around. So I would say that water, temperature, climate change, and associated pests and diseases will be really, really, really major threats in the future.
Yeah, well, I think we have a question from Abdullahi from South Africa. Sorry, I got it wrong. I was scrolling down. Lidabo from South Africa. He was also commenting and questioning in this direction. He says that in Africa, it's common to have very long droughts and lack of water. If we see that it's not possible to save water today, how is it going to be possible tomorrow? What do you think about, maybe is there any other opinion about specific species that you can grow in specific areas? So it's very important to grow what it's best adapted to each region. What do you think about? Do you have any opinion about African agricultural, let's say, the techniques, or is there any future perspective that is better than today? Is it possible to see a better, brighter future in this way?
We have to have hope, of course. First, we have to base our hope on science and science developments. And I think we can use the water in a smarter way. But of course, we are all aware that the population in Africa is increasing a lot, as I shown in the beginning, and the needs of water are incredibly high. And in Africa, the amount of water needed to feed the cultures it's also high, but it's important. So I think the way we can do it is to have a more efficient process. We have to think about having the agriculture more sustainable with increasing the efficiency of the processes, cooperating more with biodiversity to select the best possible crops for each area. That is I think it's the most important thing. And for instance, avoiding, because there are also the risks of pests and diseases, and avoiding the monocultures and having more biodiversity will have much more resistant and resilient agricultural fields. And sometimes we forget about that. If we just fulfilled our area with monocultures, actually my field of action in research is its impacts, functional impacts of biodiversity loss. It's my major area of research. And what we have been observing in all systems, from terrestrial systems to aquatic systems, is that biodiversity is the best buffer we can have to increase productivity and to increase stability of the system. So the ecosystems become much more predictable, much more resistant, and much more resilient. Even if it fails a little bit, it will recover more strongly and more rapidly. So I think biodiversity can be really an asset. So I think we have to think about that, and to select very well the species we are dealing with. I see, for instance, here in Portugal, in South Portugal, we also have scarcity of water in South Portugal. And sometimes we forget, because we had one of the largest artificial lakes in Europe. It's here in South Portugal, Alqueva. And sometimes we forget, just because we have Alqueva, which is a large artificial lake that can irrigate agriculture, sometimes we tend to forget that we are in an area with water scarcity. And sometimes there are some projects and some investments that probably will not be sustainable in the future, because they are not well adapted to the region. So for those who make investments, have to think very well about which are the best crops and where to put them. Because of course we all depend on agriculture. Agriculture depends on water, and there is another important issue, is that agriculture uses the water, and the water returns to nature not in the best state, because it already got the influence of fertilizers and pesticides. So agriculture has a lot of impact. So absorbs the water and returns the water not in the best state, I would say, in the best condition. So, but we just need to move in the same direction to improve our practices and to deal with this condition. And probably projects like EcoAgri-Food, in which we developed products that are more green, that are more sustainable, with a lower ecological footprint. I think that should be the direction. And we are comparing products and processes to move in that direction. Also, projects like the ClimateAlert project, in which we can have a mobile app in which I can have access to the information, I can save some water and can save some money. And we can make the investments according to the trends.
We have other questions about the app, by the way. Dr. Abdullahi Orire from the Federal University of Technology in Mina, he asks, basically and very simply, how would it be possible to transfer or to adapt this application to Africa? If it's possible, what's necessary to do it? Yep. And it also mentions aquaculture, but I don't know if there is any experience with also aquaculture, maybe inland aquaculture. It doesn't specify, but if it's also possible to extend this type of approach to aquaculture.
Thanks for the question. Actually, concerning the transfer of the tool for other regions, yes, it is possible. The only thing we need is to have internet and to have data. That's the only constraint. So if we have available data from, for instance, from the European Special Agency or from NOAA or from whatever, it doesn't matter. If we have a source for the data, if we have a data provider, we can use the same concept, because the tool is very flexible. So we can have the input of data and it will be the same. So as far as we have the data, as far as we have the access to the internet, it's possible to have it operating in Africa. Mm-hmm. This was a proof of concept. It was an European project that was funded, so we had to have this proof of concept for 3 different regions, Mediterranean and Atlantic, here Atlantic Mediterranean in South Spain, and then in Central Europe. So, and it worked. So I'm pretty sure that it will work as well for other continents, other countries, other regions. No constraint with that. The only constraint is the access to data and to the internet. As far as we have that, we are safe, no problem. It's quite flexible and quite easy to adapt. So this is the baseline, and of course we can have premium and other versions and more flexible to other countries.
Well, Carlos from Brazil, he also asks if it's possible to adapt this app to the Brazilian context, and he asks if Climate Alert app is open source or if it's easy to have access to it.
Yeah, the project is still ongoing. It will finish by this summer, and we are still negotiating with funding agencies. Where I think at least this version, I think it will be open access. But of course, we can have upgrades and specific things that can be tailor-made for specific customers. But that will depend a little bit on the. But at least the version that we provide with public money, we have to say that it was funded by funding agencies and by Europe, and so of course we have to provide and to have this to give it back to our society. So at least this version will be will be open and free access. But of course there are space for different versions, and there are space for different applications. Depends on what we are, what we want to develop, but the answer is this version it will be open for sure. But for operating in Brazil, we will need to have the input of Brazilian data, which we don't have by now, and the person who is using it in Brazil can have a look on our data here in Portugal or in Catalonia or in Germany, but will not have access to their own data because our server is not having the input of data from Brazil. But as far as we want to put that input data, for sure it will be available.
Yeah, so thank you, Claudia. We have many other questions. I'm trying to, well, some of them you already answered, but Pedro Tojo also left a comment. He found your presentation very interesting. He left his contact, and he also asks, well, you already more or less answered to this, the main challenges to address in the Sustainable Development Goals in the context of the need to feed a growing population. And his question is, how do we achieve a balance between environment and food security? Well, it's I don't know if you want to try to answer very quickly. It's a very difficult question.
It is a $1 million question. But I think we have to cooperate with nature. There is no other chance. I think biodiversity in the center, ecosystem functioning in the center. So we have to cope with ecosystems and biodiversity. And if we have that in mind, because we cannot have in mind of just having everything for the next week or everything for the next month. We have sustainability is not that. Sustainability is to have a legado and to have the things for the next generations. And so I think the major challenge would be to have that in mind. And so I think we have really to cope with biodiversity and to cope with ecosystems. I think there is no other chance. And of course, we have to use models to make projections and to make predictions. And the best models we have, the best we can adapt our actions and our strategies. For instance, when we started the project, when we used the satellite data, we had a resolution of 300 meters resolution. Now we have 10 meters resolution, for instance, for vegetation indexes and to watch from the sky with our big eye, the satellite eye, the state of the vegetation. This was a big improvement, and things are science is developing a lot, and models are developing a lot. So I have the hope that with the help of science, technology and innovation, we can be more sustainable and more efficient in the future. I think we have to improve our efficiency as well.
Well, there is also a comment from Stephanie Race. She asks if you are collaborating with other scientists in other geographies. She leaves also her comment, and she has been also developing tools similar to these ones to UK, Africa, and US, Canada. Are you collaborating with other scientists in other geographies?
Specifically in this context, not, but it will be very, very interesting. So this could be an opportunity. We have other projects not mentioned in this talk that we are collaborating all over the world with China or India or Brazil or US or Canada or Africa. But in these specific projects, in these two specific projects, no, it has been conducted very at a European scale. But that would be a very interesting challenge that we would like to move into that direction. So if you see any chance or any of the present see any chance of collaborating, please contact us and we'll be very, very, very happy to cooperate and to find for platforms of collaboration. That's the IBS, that's the IBS DNA. The DNA of IBS is to have multidisciplinary teams and with different ideas from different parts and to cooperate all together and to have a strength on that direction, because if we cooperate, I think we can move much faster than if we are just alone thinking about our big problems.
Sorry, we have another question from Tower in Ghana. I also addressed this in your presentation, but she asks about the warning forecasting features of the application of ClimaAlert. So can you explain a little more which type of forecasting or warning or maybe decision-making support that the application gives?
Okay, so we have two different tools. The mobile app has, you can have access to temperature, soil moisture, precipitation, vegetation indexes, water available in the plants. So this, I would say that evapotranspiration, but you can select one or the other. So they are all available, but you can click and select whatever you want. And you have data available for from the past, from the present, and some predictions. Of course, for instance, vegetation index, you cannot make predictions there because it's based on satellite information that we cannot make predictions. But for the others, we have the predictions incorporated. But of course, if you are aware of how your crop and our area of interest works, you have an idea about how was last year, how was my productivity last year or last month or whatever. And you have all the normal situation, you have the historical situation, the normal situation for the month or for the year, and you can compare with the current situation. So that is the way you can make a decision about, okay, I'm in the situation now is we are in a scarcity period, more than usual, and that is very easily informed from the graphs or from the maps, because we also have color maps in which people can compare with the situation. But that is for the next day decisions, I would say. And then we have to have more longer term decisions, seasonal forecasts. Of course, all of us have to be aware, I said that before, that there is a lot of certainty, but that's the best models available at the moment. I'm sure that they will improve in the near future. But we have seasonal forecasts, so we can have an idea about how is expected to be temperature, precipitation in the future, and compared to what we had in the present or in the past. And because I can select my area of interest, I can have a high resolution in my farm or in my fields, And and that's the way we can compare the data. That's for the mobile app. And then for the web tool app, we are more focused on ecosystem services provision. So we are more concerned about how is the water stock, how is the water provision in my river basin, and knowing that around the river basin there is a lot of agriculture investments and agricultural fields. So, and how is it today, and how will be in the future? And we provide that information related to water quantity and related to water quality. So, for instance, nitrogen exports and nitrogen concentration now and in the future. And we make projections with the best available data for the next 20 years and for the next 40 years. Of course, the projections can be adjusted, but it's the best available data and models we have so at the moment.
Okay, so another question from Angola, Domingos. Actually, he did a sequence of questions. They are quite interesting and provocative, and it would be nice to have your opinion about this. Firstly, he says that bioeconomy in general, it seems to be a very localized. So how your work can be replicated in Angola, or in other words, how can this approach be useful in the context of Angola? I think we already discussed a little bit, but maybe can give some more hints. Because next and in the following question, Domingos also raises the question about how to select the most productive crops. You mentioned that it's very important to follow what is local, right? And what is the local tradition, and but maybe you can give a little more input on this. So how to select the most productive crops that support droughts and at the same time do not fall dependent on other countries, or even worse, on multinationals, because perhaps there is also some lobbying and some interest here in also, and it would be nice to have your opinion on this. And yeah, this is the question.
It's a challenge because we know that the lands are not in all over the world. The lands do not have, or even within a country, not all areas are equally productive. So that is a challenge. We know that there are some areas that are much more productive than others, and there are areas that are already very arid or semi-arid, or even the desert. And it's very important to be aware where we are. And of course, I would be very interested in improving the way of producing food more locally and consuming food more locally. That will reduce a lot the footprint of food production. But as we said before, there is the risk of, if we live in an area that is not very productive, I understand that is a big concern. But of course, we have to adapt a little bit. That's why people always settled around rivers and around the coast all over the world, because we all depend on water, not only for agriculture, but also for other activities. If we see our distribution, human distribution all over the world, it's always around rivers, lakes, seacoast, because we really depend on water. But how to select the best crops, of course, that will depend a lot. So that is with local knowledge, respecting the local and traditional knowledge, that is very important, and maybe validating some, because sometimes there are also some myths, some false ideas, and we have to validate that with science. And more there are adaptation of some populations from the same species, but some populations that are better adapted to drought. And there are nurseries nowadays, a lot of nurseries of new plants and new crops better adapted to drought or to higher temperature. That's a big business nowadays, but probably will depend on that, all depend on that in the future. I also see that in Portugal. South of Portugal was one of the best wine producers. Nowadays, they are always investing in the north. They are also investing in higher altitudes because of temperature. And most probably they will start to invest in England or other countries because we have to adjust. But things are dynamic. Things are dynamic, but we know that things, yeah.
Maybe there is another question here that also can take some of that answer, and maybe you can add some more thoughts on this. Selma from Namibia, she's asking if you see how to use or to incorporate any way to incorporate indigenous knowledge in your project. So how would it be possible to include or incorporate indigenous knowledge, so basically traditions or habits in this project or in this framework?
Yeah.
Additionally, she's asking if you're using citizen science to improve your data somehow.
Yeah, thanks for the question. Very important topics, very, very important topics. Of course, we have to talk with people and interact and engage stakeholders. We have to listen to people. And that is very, very important. People in doing science at universities and institutes cannot be independent from people. So citizen science is here really important, and listening to the people and learning from the locals. It's very important. It's for sure that is very important to introduce. We have incorporated that a little bit in these two projects, but I think we should do that more and more in all our projects related to these topics, water and agriculture. If we don't listen to people, if we don't take into account the traditions and the local knowledge, it will be a lost. And so yes, the answer is yes. We have done it, and we want to do it more. Yes.
Okay, so one final question. One final question from Karen Bermuda. She's asking, well, in the case of small developing islands, well, she's surrounded by water, but it's salted water. Do you see, how to address water resources in this case? Because in their case, also water resources are very limited, although they have all this salted water. Is there any insights on this already?
Yeah. Yes. What I can come back a little bit, and for instance, in South Portugal, as I said before, because we have water scarcity, what we did is we introduced an artificial lake in which it's a stock of water for agriculture, and it was the main idea of doing that. But for instance, in other regions, what people have been doing, for instance, Israel is one of the countries that have been doing that a lot, but also Spain have been doing that a lot. But Israel, I think it's a very good example of that, has been using desalinization. So stations in which desalinization is used for to get the water needed for human consumption, irrigation, or whatever. Of course, a few years ago, it was really expensive. It was a very, very expensive investment. But nowadays, it's getting the prices are getting lower and much more efficient. So I think for an island with water scarcity, I think that should be the solution. And even in all countries or in all areas, one thing that we should also be aware is that it does make sense. Some countries do it, but it does make sense to use water from the company to irrigate water, and some countries do it. So it's totally unacceptable. We have to reuse the water. We can use the water that has been used for other uses, and use it for irrigation or for cleaning the streets or for irrigating the public gardens, etc., etc. It's also a good strategy. But for the specific question of those who are living in islands with water scarcity, I think desalinization is the answer. Otherwise, they will get very dependent on importing foodstuff.
Mm-hmm. Well, we're very quickly approaching the end of this session. Maybe in the future, it's going to be possible to have some bio-based solutions by IBS for desalinization, for example. I don't know, it would be very nice because it would be much more sustainable. So in these last few minutes, maybe if Felipe or Claudia would like to address the audience with some further thoughts about how to interact with IBS or interact with you, to leave a final message, it would be nice. Thank you very much for being with us today. It was very interesting discussions. Felipe, maybe if you want to close.
Okay, thank you very much for attending our session on about IBS research, and I just placed in the chat a link for a bioscan video in YouTube if you want to see more about it. And of course, I'm available. Anyone that wants to contact me. I also displayed in the last slide our Twitter from our research group if there is any need to see what we have been doing most recently. And basically that's it. I'm available and interested in collaborating, so you are welcome to contact. Thank you very much.
Thank you, Felipe. Claudia?
Thank you all for all your attention during these last 2 hours. It was very enthusiastic for me, and I'm glad that you were with us. I want to thank the AIR Center for this networking, Friday networking. That is very, very important, very challenging, very nice. And of course, at IBS, we are very open to cooperate with you guys. So please email us and contact us, and we will try to find a way of cooperating in a transdisciplinary and in a transbordery way. So thank you very much.
Okay, thank you, Claudia. I also thank everyone to be with us during these 2 very interesting presentations. It was a very big pleasure for me to be with you today. Big pleasure to be with AIR Center also. I would like to thank AIR Center staff, Moutinho, and all the others for allowing us to be here today. I hope we can be back in the future with other interesting discussions. Hope to see you soon in the future. Thank you very much, and all the best from IBS. Thank you.
Thank you very much, everyone.
Thank you. Thank you.
Bye.
Bye-bye.